Hey, I was listening on. What up, is Rutherford here. Just finished a workout, cleaning out my desk. You know, it's spring break, so we spring cleaning. Got to take time to catch up on stuff while I'm feeling super motivated and have the extra time, you know, because life's busy. Got to take care of my students. So I'm recording this video here, like a bunch of videos when I'm on a roll, and it's getting easier the more and more I do them. You know, I was worried about starting these. I didn't know how much time it was going to take. And, you know, all these like things that I was thinking way too much, just got to sit down, got to record a video. You know, it's better than nothing. Okay. I'm not here to be perfect. I'm here to work, you know, give it a shot. So we've got Unit 5 out here with us, and let me screen share this business and get it set up so we can talk about some stuff. Now, Kalia just texted me and asked about the study guides you got to work on. So, you know, girls trying to catch up on spring break, you know, that's what spring break is for, you know, catch up on what you need. And sometimes that's not just a bunch of relaxation and stuff. Okay. So like, I also remember that she and maybe some others asked me about this African violet flower color, you know, and it's like, it's springtime. So I'll be planting some strawberries, bought them from the store. So I'm going to do this one and then we'll get into the evens. And I only have 30 minutes before I got to leave. So I'm putting the pressure on, and we know when there's pressure, that's when you got to structure your day. You got to have time limits and you got to have pressure. Otherwise, most of the time, most people don't perform, you know, unless your name's Gabby. An African violet grower observes genetically identical African violet plants in the walls of the greenhouse that have white flowers. Plants farther away from the walls have blue flowers, and that plants going nearest to the center of the greenhouse has dark blue flowers. Okay, so like, what are you thinking here? They're all mating together. It's a freaking greenhouse, you know? Like if you can picture a greenhouse, maybe this requires you understand as a house, you know? They're all in the same spot. So the bees that are coming back and forth to pollinate these, and they're planting and stuff, you got to imagine that everybody's got the same genes, but we have different phenotypes. Okay, so Unit 5 is genetics here, and if we have different phenotypes, that's an environmental impact. Okay, so they got the same genes. Let's look for the questions. Warmer temperatures result in genetic greenhouse in general. It's all the same temp, you know. Plants along the walls of the greenhouse are homozygous. No, they don't have different genes. I told you that's not what we want. An enzyme responsible for flower color does not fold in cooler temperatures. Again, it's not a temperature thing. More light is available along the walls of the greenhouse. Thank you. That is an environmental. Okay. So I'm feeling good about that. I'm just going to roll it. Oh, and I'm wrong. Okay, no problem. So we got to get excited when we get something wrong, because that's an opportunity to learn. Okay. An enzyme responsible for flower color does not fold correctly in cooler temperatures. Okay. Enzymes do need the correct temperature to fold. Okay. And the greenhouse is warmest in the center. Okay, so I'll agree with that. Okay, in a greenhouse, yeah, if you're right, we know if you're right next to the window, even if we got air conditioning in here, if you're right next to the window, it's going to be a little hotter. And if you're farther away from the window, okay, I'm okay with that. All right, I jumped the gun. I'm a little excited, but hey, that's perfectly fine. We're going to keep moving on. All right, number 2. Okay, I'm going to go straight to the questions here. Which of the following best explains the cause of the phenotypic variation observed in butterflies? Again, if the phenotype is different, chances are there's just some environmental factor. They could have the same genes, but they got different phenotype, you know, just like tanning, you know, skin or whatever. Different mutations occurred in the caterpillars. Again, we're talking about different phenotypes, not different genotypes. So when they use the word mutations, I'm like, that's genotype. So I'm going to look at phenotype. I'm going to write physical protein or something. I'm just not likely to think it's B or A. B says the energy used to grow a larger body results in butterflies with lighter colored wings. Maybe. I don't know. That doesn't typically make me think of that. Let's keep looking. Individual caterpillars. I'm trying to shortcut this, right? Like obviously I could read the question and then go to the stuff, but shortcutting. Individual caterpillars evolved adaptations. Wrong. No. Evolution shows us individuals do not adapt. No. A population adapts. Some have it, some don't, and those that don't have it struggle. Those that have it, that's the adaptation. And so the genes are passed on. genes are adaptive, yeah, not individuals. I can't even keep reading number 3. That is just, this is a evolution memorization. Individuals do not evolve. D, there is differential gene expression. Oh, different gene expression. Gene expression, that's phenotype. So I'm really liking that. If I'm going fast, which I am today, bing, bang, boom, I like D, okay? So like, I'm not even reading the rest of that business, but like, I'm feeling pretty good. So I can go back and try and explain that now if you want some more explanation. Gene expression of wing color and body size in response to colors of light, so different environments they were exposed to. So you see that with caterpillars exposed to red light and blue light, the wing color is darker versus lighter, and the body size is smaller versus larger based on the different lights. Okay, grown in similar cages, exposed to different colored lights. Great. So differential gene expression in response to colors of light that the caterpillars were exposed to. I mean, that's just like, they're just asking if you can read this table. Just says red light, blue light, you know, and dark, small, light, large. That's just, that's pretty basic there. Sometimes you don't really have to think too much about these problems. If it's like, yeah, there's a different color, so the answer says different color, you know, again, here I'm zooming in on specific words. There's all this stuff here, right? But the words that I'm zooming in on are phenotype, red, blue, gene expression, and colors. I mean, it's like, you don't have to know much more than that. They're trying to make it complicated when it really ain't. Okay, next one. Oh, fun. All right. So now I'm feeling potential offspring with no error, potential offspring with error, possible offspring of female. Oh, so this is a Punnett square kind of a thing, maybe. All right. How would an extra chromosome affect the male offspring reproduced by gamete? So first we got to make sure that XY is male, because that is for humans, but yes, are we talking about humans? Yeah, okay, I guess we are, right? And so if we talk about red-blind colorblindness, which we have, we know that it's on the X chromosome, and it says it right there again. Like, we think we have to memorize all this stuff for the APbio test? Not really, okay? They're telling you right there. It's associated with the X chromosome. Now, if you've heard it before, then it's like, okay, easy. Like, I've heard this, I've taught this. So I'm like, boom, I know what they're talking about. Otherwise, the information's right there, okay? So they're asking us, how would an extra chromosome affect the male? So they're telling us, okay, if we have an offspring, here's the female, XX, here's the male. This is a Punnett square. Again, if you've done Punnett squares, this will be really helpful because they're not telling you it's a Punnett square, but you can look at this and go, okay, so there we go. Big B is full color vision and little b is red line colorblind, which I told you, you don't really know exactly what letters they're going to use for this stuff, but they'll show you, they'll tell you the letters, and here they are telling you right there. So here we have a carrier female, here we have another carrier female, here we have a male who's colorblind, and here we have another male that is colorblind. So all the males coming out of this one are going to be colorblind. And then over here, wow, they're saying if there was a mutation and you got an extra X, oh, this male here, this is a very weird thing. So when this guy makes this sperm, some sperm has no chromosome of X or Y. Probably all the other autosomals, 22, are fine, but here they've got the X and the Y, so it's like non-disjunction. And then you've got these males over here that just have an extra X. To me, that means, wait, they have the instructions to see color and not see color. Big B and little b. See color, not see color. So I'm thinking that if that actually comes out to be, like, if that can be born a human being that functions, which it can, and this one, that also can be born a female who's like kind of not super feminine, only with one X, that can be born because the other 22 autosomal chromosomes, they still have like a normal pair, presuming that they do. But here I'm thinking these guys can see color. They got that capital B, okay? So the male offspring would all be colorblind. Nope. The male offspring would full color vision. I mean, I like C. That's just straight up what's going on, okay? Just like these females over here. If you're hetero, you can see color, no problem, okay? That's what we know with the ladies, and it even tells you right there. A female who's red-blind do they tell you that this is full color vision? I don't think they tell you that these females are full color vision, so you just have to know it's a carrier, and that this capital B is dominant, which means that as long as you've got the instructions in one spot, you're good. And so I like C. Could be, yeah, okay, great. Don't even have to look at the rest of them. Nice. Number 6. Scientists have found the DNA methylation. Okay, which is putting a methyl, which is CH3 or CH4, adding that little thing onto DNA will kind of like, that's a little lock. And then the DNA can't be transcribed or translated, so no gene expression. It suppresses the crossing over in the fungus. Which of the following is most appropriately raised? Is the level of genetic variation related to the amount of DNA methylation? Well, more locks. Okay. Without crossing over, will gametes be viable and be able to produce a zygote? So what we know in crossing over, this requires you kind of know crossing over, is that crossing over increases genetic variety in meiosis 1. It's not a necessary thing at all for like sustaining life. We have plenty of ways to get variety, plenty of ways to evolve. This is one way, so it's not necessary. So B, I'm not liking. That's not an appropriate question. We know that they're going to be fine. C, does DNA methylation result in shorter chromosomes? No, it's a lock on the DNA. It doesn't shorten the chromosomes. Is this species of fungus a diploid organism? I don't think it has anything to do with that. So I do like A, because maybe if DNA methylation suppresses crossing over. So what you can do right there is you can write lower genetic variety, okay? Less evolution, less genetic diversity, less variety. I don't think it has anything to do with diploid. It doesn't. I like A for that. B, it suppresses crossing over, but yeah, A is so right. A just literally says the level of genetic variation, which I said is that's what you need to think of when you think of crossing over. Genetic variety, lower genetic variety. So I got to go with A. It's real strong. Yeah, buddy. Number 8. Come on now. Read each question carefully. That ain't me. Oh, okay, yeah. So in free response, which we'll go over later. I'll make some more videos on that. Don't you worry. Okay. I ain't worried. You ain't worried. There, let's keep moving. Pedigree. Okay. So again, they're telling you exactly who has cataracts and who doesn't. Here, you just got to know males and females and who gets married and parents and all that stuff. And they're just seeing like, maybe guessing, do they tell you whether it's, first thing I'm gonna look for, is it dominant or recessive? Because they're not really telling you. Oh, look, and you see the answers down here. I knew to look for dominant recessive because that's like, I'm kind of like, ooh. And down here it says recessive X or recessive, dominant X or dominant, okay? So that was X linked, sex-linked chromosomes and traits. That's gonna be like your color blindness and stuff. So whereas your somatic chromosome, that's any of the other 22 human chromosomes that are not sex chromosomes. The sex chromosome is number 23, X and Y. All right, so what do we think? This one, man, let's look at this couple right here. Okay, male don't have it, and the female has it, and boom, daughter has it. Okay, 2 parents, only one has it, boom, have it, right? However, these 2 parents don't have it, and then their kids get it. So how do the kids get it if the parents don't have it? You know that is recessive. You know it's recessive. Because that's like your 75-25 kind of setup. Yeah, obviously when we got like double recessive here forming full recessives, okay? So I know it's recessive. Do I think it's somatic just in general, but if we go for the X chromosome, that means whenever you have a sex-linked trait, it is going to show up more in the males, like colorblindness. That's not what we see here. We see, look, we got 1, 2, 3, 4, 5 females that have it and 3 males that have it. Okay, so that's not sex-linked at all. I've got to go with B. Boom, Shazam. That's my answer. Okay. Sweet pea plants, which I'm growing in my yard, had some today. Okay. Purple flower color is dominant. Interesting, because all of mine are white. Over red flower color, none of them are red. Anyway, long pollen grain, Sweet pea plants are heterozygous for flower color pollen. Boof. Okay, this is a double punnet because we are talking flower color and pollen grain shape. So you can kind of, these ones are going to be good to like write this down. So this 9331, that's a double hetero. Okay, so you can just kind of remember if we have a double hetero, we're going to go 9331 in the phenotype ratio, what they physically look like. Okay, and so that means that the 9 is going to be dominant, dominant, and the 3 is going to be dominant recessive. The other 3 is going to be recessive dominant, and then the 1 out of 16 will be recessive recessive. That's what we're going to see here. Okay, so which of the following statements provides the most reasonable prediction to account for the deviation from the a deviation? What is it saying? I got to go back to the question. You got to look. 2 plants that are heterozygous. Okay, a geneticist surprised to observe that there are far fewer round purple offspring and long red than they expected. Far few round purple, long red. So now I'm going to write down real quick what's dominant and what is recessive. So dominant is long, and then the recessive is round. And then for flower color, purple, dominant is long purple, and then recessive is round red. It's nice and easy to remember. Round red is recessive. Round red recessive. I'm trying to make it easy on you. Okay, so they said there was a very low amount of round purple, which is the 3, and then and the long red, and long red is the other 3. So what they're showing you here, which is like a classic thing, if we have more, like we have heterozygous, but most of these bad boys are either dom-dom or recessive-recessive, and there's not very many of these mixes. What that means is that on the actual gene, on the chromosome, the gene, flower color and pollen shape, the little genes on the DNA that encode for those proteins, they're very close together, okay? If the genes are literally farther apart, then they can cross over and they're not really linked. This is called linkage, not sex linkage, but these genes are linked because they are so close together. So when one moves, the other moves. If it crosses over, they're going to cross over together. So we're seeing this word linkage right here. I bet that's our one. Genetically linked. Yep. So that's just a classic linkage thing. This is just kind of a memorization thing where they're giving you this ratio. They're telling you what's dominant recessive. You don't have to have any of that stuff memorized. All you need to know from this is if you get a lot of individuals that are dom-dom and a lot of individuals that are recessive-recessive, and you don't get a lot of like weird mixes, then the genes are very close together on the chromosome. That is called genetic linkage. We roll it. 14. Okay, mitosis, meiosis, parent cell diploid. Mitosis might result in daughter cells with different numbers of chromosomes. Well, mitosis, you just go through PMAT once, and then meiosis, you go through PMAT twice. Is this the right question? Okay. In mitosis, chromosomes consist of a single chromatid. No. In mitosis, synapsis of homologous synapses, like joining of homologous chromosomes, result in 4 haploid? No. Mitosis only makes 2 diploid. and meiosis. Okay, so mitosis produces one identical daughter cell. No. Mitosis produces two identical diploid daughter cells. Hell yeah, that's just a definition. Meiosis produces 4 haploid. Hell yeah, that's just a definition. Shazam. The rest are just trash. This is just memorization of mitosis, meiosis. Kaboom. Okay, 16. Ooh, data. Okay, so F1, F2, and the P, parent generation. This is how we take grandparents to parents to kids in genetics, Punnett squares. So 2 mouse genes, fur color and fur length. Okay, so again, I'm going to write down dominant or recessive. Always keep that spare scrap paper. Black fur is dominant over white fur is little b, and then long fur. So black long is dom, and then short fur is little l. Okay, so I'm writing black long and white short. Good, and they cross them, and 2 true breeding mice. So true breeding mice are going to be the ones that with long black fur and with short white fur. So they have the dom, and then they have the recessive. So this is similar to that other question we just did. Their progeny, the F1, all are hetero genotypically, but their phenotypes obviously are going to be dominated by the capital B, the capital L, which is black and long. Okay, great, right. Simple stuff. Yeah, if you notice this one, like I got to read the problem because I'm going down here going 1, 2.2, 2.4. I'm like, I don't even know. Which of the following is the mean number, the average, per cross of F2 generation offspring that are a result of crossing over? Oof. Like I kind of like that last one that said, if the offspring look like the parent, there's no crossing over. If the offspring look like the dom parent or the recessive parent, there's no crossing over. But if you get someone who's like, oh, I'm making black fur, but short fur, that's a dom recessive. Or if you're making white fur that's long, you've got a dom recessive, like a lowercase b, capital L kind of a thing. So that's what we're looking for here. And the average number that result in that. So the total number of F2 generation phenotype we've got right in the middle is the long white and the short black. Those are the weird ones there, okay, right in the middle. So we're looking at, for cross 1, there was 1. For cross 2, there was 1. For cross 3, there is 0. For cross 4, there is 2. For cross 5, there is 2. Well, we've got to add up these middle ones. Okay, so they're asking for any of the ones in the middle. Okay, so for cross 1, there's 1. For cross 2, we're adding up these 2 middle numbers here. And so there's 3. And then cross 3, we're adding up these 2 middle numbers. That's 1. Then adding up those for 3 and adding those up for 3. So my mental math tells me that that average is going to be somewhere around 2. I mean, I don't know if it's 2.2 or 2.4. I mean, I do know that it is going to be 2.4 because there's 3, 3, and 3, and then 1 and 1 are going to drop that down. So I'm feeling comfortable with 2.4. This question was a harder one. There's a lot of steps in that one. Okay, 2.2. So I should have actually done the math. I could have added those up pretty easily if I would have gone like, 9, 11 divided by 5. How many times 5 go into 11? 2 point. I should have checked 2.2. Okay. Yeah, there's quite a bit of knowledge that you have to have here as far as that. But again, notice it's the same as that question before. Once you know what crossing over does and genes being close to each other and being able to move, then they're going to tell you what's dominant recessive. You just have to know that those weird hybrids that have a bit of dominant and a bit of recessive, those ones are more rare if the genes are really close together. They're linked, okay? All right. So Z-dub for birds, ZZ is male and Z-dub is female. So they're telling you right here, this is different than human beings, okay? So don't get it twisted. The gene for feather color is located on the Z chromosome. So, males and females get instructions, but the male has 2 instructions for feather color. The red allele is dominant over the brown allele, okay? So, and we know that it is sex-linked, okay? So if you have a brown male pigeon, brown is recessive, okay? It says it right there for brown recessive. So what letter are we going to use? We're going to use R's because red is dominant. So we use a capital R for the dom, and then I'm going to make a little freaking Punnett square. Hell yeah, first one over the brown allele. So it says 3 crosses between brown male and red female, okay? So I'm going to draw Z with a little r, Z with a little r. That I'm labeling male or dad, okay? Then we have Z big R and dub for the red female pigeon because red is dom and it's on the Z. I'm just double checking myself here. Okay, great. And then, okay, great. So what are the following is the average number of male offspring produced by the 3 crosses? Male offspring? Wait a minute. It ain't even asking about color? Oh, you already know. The average number of males is half, okay? Half, okay? That has nothing to do with colors. Like, so in cross 1, they're having 20 babies. In cross 2, they're having 20 babies. In cross 3, they're having 20 babies, okay? Which of the following is the average number of male offspring produced by the 3 crosses? Oh, maybe we do have to... Gosh, I mean, it's got to be 9 or 11, because like it's right because you just got to have half males, half females. This is so. I guess maybe we do need to know the colors here because it's 9 or 11, but maybe we can kind of see. All right, so the in cross 1, gosh, this is so interesting. I just don't really feel like. What are our chances? I'm going to draw this out and see if the actual Punnett square helps it make sense. Okay. And I think it does reveal that all the females, every single female is going to, this does reveal it. So you do have to have this Punnett square and this knowledge. Every single female is lowercase r, which is brown. Okay. Every single female is brown. And then up there, actually, every single male is heterozygous red. Every single one. So all of the red boys here are boys. So the red goes 11, 9, and 7. The average of that is 9. Final answer, D. Hell yeah, let's go. Okay. More of the business. Let's go. So we know these genetics ones, they do take a little more time. You got to do some math, some calculations, or draw some Punnett squares. I've had to write the most on this. So when you find a Punnett square, know that you're going to, genetics problem, you're going to need a little more time, and that's fine. Okay. Then we race through the other ones that are easy. Okay. Fruit flies, we know that. Purple eyes, ebony body are traits of autosomal recessive. Okay. So purple and the black or ebony is autosomal recessive. They're asking, do you know that wild type is dominant? Okay. And wild type flies are going to have the dom. Are they telling you what? Normal. Okay. So wild, I'm writing wild, dom is normal and normal eyes, normal body. Okay. So normal. But then the mutant recessive is purple and ebony. Okay, so we might be using N's because they're talking normal here, but I don't have to do a Punnett square. They're going to throw this vocab word at us, chi-square. Okay, we haven't gone over the chi-square math yet, but we'll get that sorted out. It's pretty simple. Okay, so woof. This is hard to explain without telling you exactly how this works because you got to set up some math for this. It's pretty simple. Observed minus expected squared over your expected. So they're telling you observed and expected. This is that, like, if you did a Punnett square and you just followed the ratios, you see these expected numbers. That's 9, 3, 3, 1. That's the ratio you got to remember for 2 traits in a double Punnett, 9, 3, 3, 1. And so we're expecting they have, you know, a bunch of babies, right? And they're like, okay, well, we would imagine that we would have 171, 57, 57, and 19. That's what we expect based on the probability, which comes from the Punnett square. However, we're observing, again, this is the third question like this. Come on now. More of the dom-dom and more of the recessive-recessive than we expected. Okay? So likely, and it's not a lot more, it's a little bit more. So likely these genes are somewhat close together, but not insanely close. Like if this said like 200, 20, 20, 40, then we might be thinking like, okay, these genes are very close together. They are linked. But you got to just do the freaking calculation on this. This is not one that I can easily explain on video. This is something that we've got to do, and you got to look at the actual graph there. So we could just find the answer. Yeah, this is just something you got to calculate. So the answer is D there, but we'll do those calculations. That's a in class kind of a thing. Okay. Great. 22. The dragon lizard, the Z-dub sex, male is ZZ. Okay, so lizards like birds, chill. Okay, all the eggs incubated at temperatures above 32. So this is a thing with like frogs and lizards, some frogs and lizards, that like temperature determines their sex, which is wild. Okay, and not fully, but it says tend to develop in the females. So which of the following best explains, we got a best explains here, how the development of phenotypic female dragon lizards occurs when incubation temperatures are above 32. Okay. So they're basically telling you something and they're saying why. Okay. Interesting. So let's see what it is. The males are ZZ, but it is more likely that that the females develop when the temps are higher. Lizard embryos with ZZ cannot develop above 32. No, they're saying tend to. Okay, it's not that there's no males when it's hot. B, an incubation temperature above Z chromosomes are mutated into dubs. That would give us more females, but that's a very weird concept to think that like an entire chromosome could just like mutate into another one. That's like freaking weird. C, an incubation temperature above 32, crossing over transfers genes from the Y to the Z, producing females. Nope, because what they're telling us is that your ZZ individuals might become females. So the ZZ, the genetically ZZ, they don't even have a W chromosome there. They don't really like C. D. Hopefully D is nice. Incubation temperatures above 32 inhibit the genes on the Z chromosome that produce proteins necessary for male development. There it is. There it is. If you've got the Z, but it's too hot, then it is going to be inhibited. And then in the absence of being male, maybe you just be female. So this is something that we know. We've got X chromosomes and we've got Y chromosomes. As human beings, you boys have the X chromosome. We know how to be girls, but the Y comes in and says, no, we're going to make some testosterone. We're going to override the female. But guys know how to be girls. Girls do not know how to be guys. It's the opposite here with dragon lizards, but it's the same concept. Males are ZZ. They only know how to be males. Females, however, know how to be males, but they also know how to be females. So the female knows both. So for D here, we're saying, all right, if we chill out that maleness, Yeah, maybe not. I don't, because if you've got a ZZ and it chills out the Z chromosome for male development, you still don't have a dub. But honestly, this is where in a tough question like this, you got to like not just be like, well, I don't know, I have no freaking clue. You got to like chill out and think D is okay. The other one that I liked, I really didn't like A. A is total trash. C, I think B said an entire chromosome mutating. That's like really freaking weird. C says crossing over transfers genes. But this is about egg incubation. You know, I think what they're testing with C is that meiosis is already done. Crossing over happens in meiosis, but once the egg is laid, mom is not making the egg or dad is not making the sperm. C can't happen at that point. The egg is already laid. And now if the egg is incubated, so B is an okay answer. I do think D is the best. I'll be shocked if they say C here. There we go. D. D is the best for reasons discussed. Cool. Always love a challenge. Hell yeah. Always look at the tough questions and be like, let's go. Trisomy 21, extra chromosome pair 21. This is Down syndrome. And okay, age of mothers at birth, primary cause of the error leading. This is going to be non-disjunction. So this is something just to memorize that Down syndrome, trisomy 21, happens with older mothers giving birth to their babies, because as the mothers get older, you have a greater likelihood of malfunctioning and in the meiosis process. And non-disjunction, which is to occur in anaphase 2, if there is, and maybe it can happen in anaphase 1 too, but non-disjunction is when you're supposed to split them apart, but they pull, they tug of war, and it just doesn't break. And so then you can have 2 chromosomes in this egg and no chromosome in this egg. And so trisomy 21, you're only supposed to have 2 chromosome number 21, one from mom, one from dad. But if you have 3, your mom gave you 2 because her ovaries made a little bit of a mistake there. Yeah, okay. So we're looking for older moms, and we're looking, yep, okay. This is just, you got it. Like, if you remember trisomy 21, Down syndrome, non-disjunction, and you can picture that in your head, there's no reason for me to look at any other thing, okay? We've got non-disjunction. Oh, we've got non-disjunction in every, ooh, that was a good check. I almost just clicked A and moved on. We have the word non-disjunction in every single one. Great, okay? At older maternal ages, older moms, there's an increase in the number of errors during mitosis. Not mitosis. Okay. The incidence of non-disjunction errors in meiosis, yeah, during sperm production, it's from the mom. This is older moms. It says it right here, maternal age group, older moms. It says nothing about old dads, okay? Guys can make like healthy sperm like well into their 70s. It's weird, but that's just what we do. We do quantity over quality. Ladies have a much shorter window here. So at older maternal ages, again, back to the moms, the incidence of errors in meiosis during egg production increases. Yeah, okay. So I do like C, but I'll check D. Errors in meiosis leading to non-disjunction are more likely to occur during meiosis. Oh, that's nothing to do with it. D has nothing to do with it. We're talking about old moms here. It says old moms. Okay. Now you could just know this from reading the graph. You could figure this out from reading the graph. But if you memorize this whole concept of non-disjunction in old moms and Down syndrome, then you would be able to answer this question a lot faster. So just know with most things here on the APbio test, if you study really, really hard, most of these questions are just going to be easier. But if you don't study or you don't know something, it doesn't mean you can't answer the question. It's just going to take a little longer. Okay. Maybe like a little more brain juice, which is chill. What's this C? Let's go. Let's go. Uno mas 26. Oh, too easy. Just like one line. Get out of here. Okay. I'm going to go to the answers. First division. will reduce, move, reduce, result in crossing over, reduce the number of chromosomes, move single chromatids. Woof, gotta read the question now. A diploid yeast species, bacteria, prokaryote, can reproduce sexually or asexually. Whoa. An experiment was performed that to induce mitotically dividing, which would be asexual, cells in G2 to undergo meiosis. Whoa. Which of the following best describes the steps that cells will follow to form gametes? Wow. Stand by. An experiment was performed to induce mitotically dividing cells in G2 to actually undergo meiosis. Meiosis meaning they're going from, okay, well, they were trying to do mitosis, which is like asexual reproduction, and then they move over to meiosis, which would be making a gamete for sexual reproduction. So they're in G2. So this is requiring your knowledge of the cell cycle, G1S, G2, and S phase, DNA replication. So then you get to G2, and now they're switching them over to meiosis. Which of the following best describes the steps to follow to form gametes? So I think they're just going to ask us how to do meiosis. The first division will result in crossing over. That there's no reason to believe that. I mean, it might, but that's just not what we're talking about. We're not talking about genetic variety with this. We're just talking about making gametes. The first division will reduce the number of chromosomes in half. The second division will result in each daughter cell having 1/4 of the original. That sounds great. That's meiosis. It just says we switched them over from mitosis to meiosis, and B looks great. Okay. Let's just check C and D. The first division will move single chromatids to each daughter cell. No, that's not, we can't do that. That's a skip and a step. That's not how meiosis works. Okay. You got to go through PMAT cytokinesis and then PMAT cytokinesis again. Okay. You can't skip ahead to C, that's not how evolution works. The first division will reduce the number of chromosomes by half for each daughter cell. Second division will move single chromatids to each daughter cell. I don't like that. Single chromatids. I just don't like that phrasing. Honestly, I can't describe this one right now, and I'm just like excited to be done, but resist the temptation to rush and finish at the end. I have time, so I'm going to stop, and I'm really going to look at this because every question deserves your love and time, attention, and care. Okay? So the first division, B says, yeah, they both start the same. And then right here, if this is a paper test I'm writing, the second division will result in each daughter cell having 1/4 of the original number of chromosomes, which is true. The second division will move single chromatids to each daughter cell. Yeah, I don't like that move single chromatids. That makes it sound like you've got your daughter cells and then we just put a single chromatid in each one. It's not how it works. The concept is that this vague result in each daughter cell is like, yeah, this is complicated anaphase, telophase, cytokinesis, and then prophase, metaphase, anaphase, telophase, cytokinesis. We're not moving them to the cells. I just like the wording on B a lot. Oh, man, they got me. Ugh. Yeah, gosh, I don't know what's wrong with B. Yeah, I don't know what's wrong with B. That's like freaking weird, but you win some, you lose some. Like I said, you don't got to get 100% on this test, you just got to do better than most of the kids in the nation. Lego, thanks for coming in and watching. That was Unified.